Hi folks, welcome to lesson 5 of unit 1 of the advanced herbology course. Making good progress, we're almost finished the first unit, and today's lesson is going to take us through topic 1.5, which is the protein control of cell division. So the first place we start when we're talking about cell division is in terms of the cytoskeleton, which helps to keep the structure of the cell, but it also plays an important role in the actual division. So the cytoskeleton gives mechanical support and shapes to the cell. I always think of it a bit like scaffolding within the cell. Metal bars going off at all angles to support and maintain the integrity and structure of the cell. It consists of different protein structures, including microtubules, which are found in all eukaryotic cells. Microtubules themselves are hollow cylinders composed the microtubules control the movement of the membrane-bound organelles and chromosomes. Okay, the movement of organelles and chromosomes. So they play a really, really important role within the cell, and often don't get enough credit, especially introductory levels of biology. Now, the microtubules, they tend to radiate out from a place known as the microtubule organizing center, or the mTOC. And there's a specific type of mTOC called the centrosome, which we are interested in terms of what we're looking at in this topic. Now, cell division requires remodeling of the cytoskeleton, okay? It requires the cytoskeleton to essentially morph and change its position and its shape. And the formation of breakdown of microtubules involves polymerization and depolymerization of the protein tubulin. So as the microtubules build up and break down, as we can see in this animation here, okay, we can see polymerization of the microtubule here and the tubulin been added to it. And then obviously depolymerization is the opposite, where the tubulin is removed and the microtubule becomes shorter. The microtubules form the spindle fibers that are active during cell division. And again, we can see in this diagram here, we can see spindle fibers emanating there from the mTOC. And we can see the microtubules form the spindle fibers that are active during cell division, and they attach themselves to the chromatids and help to pull those to opposite poles of the cell. And then once their job is over there, they'll polymerize and return to the centrosome. Now, moving on to the cell cycle, this is probably the most commonly assessed part of this topic. The cell cycle consists of two main phases, interphase and the mitotic or M phase. Now, interphase involves growth and DNA synthesis, and that happens via the G1, the S, and the G2 part of the interphase. Now, G1 is a growth phase, the S phase is where the DNA is replicated, and the G2 phase is a further growth stage. Okay, so the easy way to remember it is G is for growth, G is for growth, and then the S phase is DNA replication. Now, interphase takes up the majority of the time in the cell cycle, with the M phase, the mitotic phase, taking up very little, a small proportion of the overall duration. Now, the mitotic phase involves mitosis, which we know a little bit about already, and the phase that happens after mitosis, which again we've learned about, called cytokinesis, okay, or the separation of the cell membranes. Now, in mitosis, the chromosomal material is separated by the spindle microtubules that we learned about in the last slide there. And this is followed by cytokinesis, in which the cytoplasm is separated into two daughter cells. Okay, and we can see this little amoeba sisters gif here, which talks us through the things that are happening during each of the stages in the cell cycle. So we can see G1 growth, we can see S phase where we're replicating the DNA, we can see G2 phase where there's further growth, and the M phase where we get cell division or mitosis happening there. Now, mitosis consists of 4 phases, okay, 4 different stages that we need to know about. We've got prophase, we've got metaphase, anaphase, and telophase. Okay, and again, initially when you start to learn about this, it might seem a bit overwhelming, all these stages, but you will very quickly learn them and hopefully be able to differentiate between them all by the time it comes to your exams. So the acronym to remember here is to remember PMAT, okay, P-M-A-T, PMAT. If you can remember PMAT, that'll help you remember the order of the stages of mitosis. And you can see again, we've got a nice amoeba sisters gif there that talks us through what's happening in each of the various stages there of mitosis, and that might be nice to watch as we go over the content here. And again, there's a couple of GIFs at the bottom there which show you the various stages as well, and it might be worth coming back to this at some point. So the DNA will condense during prophase into chromosomes that each consist of sister chromatids, okay? We're probably familiar with that first stage from National 5 even when we started looking at mitosis. At this point, the nuclear membrane that surrounds the DNA will start to break down. The spindle microtubules will extend from the mTOC by polymerization, okay, where we add those tubulin proteins to form a big long microtubule. So they will attach to the chromosome via their kinetochores, and that's in the centromere region, right, where we join the two chromatids together. So that's prophase. Metaphase. Now, the chromosomes are aligned at the metaphase plate. Okay, we used to call this the equator when we learned about this previously. So we're going to call it at National 5 the metaphase plate. So the chromosomes align at the metaphase plate. That is metaphase, okay? Probably the easiest of the four to remember. Anaphase, as the spindle microtubules shorten by depolymerization, okay? So depolymerization is the removal of the tubulin protein and makes the microtubule shorter, and that is how we pull those sister chromatids apart. That's how we separate those sister chromatids, and they are pulled to opposite poles of the cell. Okay, and I always remind my students about a former girlfriend I had called Anna, and she broke my heart in two. Okay, we split up and she broke my heart in two, and that's how I always remember anaphase. Okay, because we're splitting apart those sister chromatids and then pulling them away, splitting them into two sections of the cell. Telophase, our last section here for mitosis. The chromosomes decondense, okay? So they become less dense than they were. Basically the opposite of what happens at the start in prophase. And then new nuclear membranes are formed around them. And again, we're familiar with that, the nuclear membrane forming from National 5. So again, we're just adding a little bit to the knowledge that we had previously. Okay, and the main thing is just remembering A, the name of the stages, PMAT, and also remembering what happens in each stage. That's the tricky bit about this. And what's actually happening in each stage is something that's pretty familiar to us. And there's just a few extra bits of terminology we have to learn in addition to what we've learned previously on this topic. Again, the cell cycle is covered by the Amoeba Sisters, who I suggest you go and follow their YouTube channel. As I always say, just watch it. It's good for the animation. There will be additional details in their videos or omissions in their videos which make it different from the content in the advanced higher course. So don't worry about that. Just watch it to get a feel of what's happening in the cell cycle. Okay, so how is the cell cycle controlled? What controls each of these stages within the cell cycle? Well, progression through the cell cycle is controlled by these things called checkpoints, okay? Now, we have three main checkpoints that we need to worry about: the G1 checkpoint, the G2 checkpoint, and the metaphase checkpoint. And their names kind of suggest where we are likely to find those checkpoints. Now, what's the purpose of them? Well, checkpoints are mechanisms within the cell that assess the condition of the cell. Okay, now in different stages, what they're assessing is going to be different. Okay, so for example, in the G1 checkpoint, which is a growth phase, we're going to be assessing the growth. Okay, so we're assessing the condition of the cell during the cell cycle, and these checkpoints will halt progression to the next phase if certain requirements are not met. Okay, so for example, if the cell hasn't grown enough in G, it will halt progression to the next stage until it has grown enough. So cyclin proteins that accumulate during the cell growth phase, okay, during G1, are involved in regulating the cell cycle, okay? So these cyclin proteins, they're accumulated during the cell growth phases and they're involved in regulating the cycle. Now, cyclins combine with and activate cyclin-dependent kinases, okay, or CDKs for short. All right, and we can see in our diagram here an example of some of these. We can see this different cyclins there, and they are combining with CDKs. That then activates the cyclin CDK, and the active cyclin CDK complexes, okay, that's the combination of the cyclin and the CDKs. They are then able to phosphorylate proteins, okay? And again, in our diagram, we can see phosphorylation happening here and over here and over here in various places across the cell cycle. And that's what helps the cell to determine and to regulate if progression is going to happen, okay, if it's going to move on to the next stage of the cycle. And if sufficient phosphorylation is reached, then progression will occur. Okay, so cyclins and CDKs combine together to form active cyclin CDKs, and those complexes help to phosphorylize proteins. And if enough phosphorylation has happened, then the progression will occur to the next stage of the cycle. Okay, let's talk specific checkpoints now. Well, at the G1 checkpoint, we have retinoblastoma protein, or RB protein. Okay, you can just write RB in your exam. And that acts as a tumor suppressor. Okay, and how it does that is by inhibiting transcription of genes that code for proteins that are needed for DNA replication. Okay, so we have to phosphorylate that RB. Okay, this RB protein must be phosphorylated, and it gets phosphorylated by the G1 cyclin CDK. So phosphorylation by G1 cyclin CDK inhibits the retinoblastoma protein, the RB protein. At this point here, this transcription factor here is inhibited, and we've got the active RB protein. But when that gets phosphorylated by the active G1 CDK, that then changes the conformation of the RB protein by phosphorylating it, adding these phosphates to it, which then allows the transcription factor to become active, and then we can proceed to the S phase. Okay, we can proceed to the S phase at that point after we've allowed transcription to occur. So that transcription factor is inhibited by the RB protein when it's active, but when we phosphorylate it with G1 cyclin CDK, then we are able to then inactivate the RB protein, which allows transcription to occur and for us to progress to the S phase. Now, the G2 checkpoint, that assesses the success of DNA replication, and any potential damage to the DNA is also assessed at that point. Now, if there is any DNA damage, that triggers the activation of several proteins, including this specific one here, P53. Okay, DNA damage activates that protein P53, and that will stimulate either DNA repair, the arrest of the cell cycle. The arrest means to stop the cell cycle, or it will cause cell death or apoptosis that we'll learn about later on. Now, a metaphase checkpoint controls progression from metaphase to anaphase, which makes sense. At the metaphase checkpoint, progression is halted until the chromosomes are aligned correctly on the metaphase plate. Okay, if the chromosomes are not aligned correctly on the metaphase plate, we will not proceed from metaphase to anaphase. Okay, we check first of all they're aligned, and second of all that they're attached to the spindle microtubules. Okay, so that's the two key things that are required before we can move from metaphase to anaphase. We need we need those chromosomes aligned correctly on the metaphase plate, and we need them to be attached to the spindle microtubules. And here's a little summary that I made up just to go over the checkpoints. You can pause the video if you want and have a wee look through all of these and just check that you understand and know all of these various things. You will need to know about the checkpoints for your exam. They do like to ask about those. Okay, and you can see there's a lot of content there that they can ask about. So make sure you do a good bit of revision and learn as much of that as you can. Okay, and the last little bit of the topic talks a little bit about when cell division goes wrong. Okay, so when there's an uncontrolled reduction in the rate of the cell cycle, an uncontrolled reduction, these are important words, in the rate of the cell cycle, this may result in a degenerative disease. Okay, and I'm going to underline that word rate as well. Okay, so it's an uncontrolled reduction in the rate of the cell cycle may result in a degenerative disease, whereas an uncontrolled increase in the rate of the cell cycle may result in tumor formation. And we can see this little diagram here shows us what happens when there's an uncontrolled increase in cell division. Okay, we know this a little bit from when we looked at it in higher human biology as well. We can see all of these cells dividing uncontrollably there and forming a tumor. Now, a proto-oncogene is a normal gene. It usually is involved in the control of cell growth or division, and sometimes it can mutate. And when it mutates, it forms a tumor-promoting oncogene. Okay, so all you need to know about that is that when it's functioning, when it's normal, it's called a proto-oncogene. But when it starts to mutate and promotes the formation of tumors, it becomes a tumor-promoting oncogene. Okay, proto-oncogene to tumor-promoting oncogene. So proto-oncogene to tumor-promoting oncogene. Okay, folks. And then lastly, we're going to look at apoptosis. Okay, apoptosis, that's the programmed cell death that occurs. Now, Apoptosis is triggered by cell death signals, okay? And these can be both external and internal. And we can see on our diagram here, apoptosis triggered by death signals. We have external signals such as a lymphocyte, or internal signals such as DNA damage. Okay. Now, external death signal molecules bind to a surface receptor protein and they trigger a protein cascade within the cytoplasm. Okay. And it's that protein cascade which is going to trigger the self destruct mode, the apoptosis within the cell. Similarly, an internal death signal resulting from DNA damage will cause activation of the p53 tumor suppressor protein. Okay, and again, that will lead to apoptosis of the cell. Now, both types of death signal result in the activation of these proteins called caspases. Okay, they're a type of protease enzyme, and what they effectively do is they catalyze or break down proteins within the cell. And that is what causes ultimately the destruction of the cell, okay? These caspases breaking down all of the proteins. Apoptosis is essential during the development of an organism to remove any cells that are no longer required as development progresses, or in other organisms during metamorphosis. Okay, when it changes from one form to another, some of those cells will no longer be relevant, and apoptosis is needed to destroy those cells. Now, sometimes in cells, they may initiate apoptosis if they simply do not have enough growth factors, okay? So, for example, at the end of the G1 checkpoint, if the cell has not grown enough due to an absence of growth factors, then the cell will just self-destruct. It will undergo or initiate apoptosis, okay? Because that cell is not going to be any good if it hasn't reached the required size. So I hope this little summary diagram here is useful as well. It's just another way to present the information. You can see that the last step in each of these is all the same, that activation of caspases that causes the destruction of the cell. But the reasons for that will obviously vary, and I've hopefully covered all of that in a nice, neat diagram there for you, and that's helpful for you as part of your revision. Okay, folks, so that's the end of topic 1.5. Hope that was useful for you. As usual, leave any comments about things that you found useful or things that you want to ask more questions about, or you're still unsure about. But for now, folks, thanks for joining me, and class dismissed.